हेलो एवरीवन, आई एम नैंसर एंड यू आर वाचिंग गो टर्टल बायोलॉजी। इस वीडियो में आपको प्रोकैरियोटिक सेल के सारे सेल्यूलर कंपोनेंट्स जो नीट के लिए नेसेसरी पॉइंट्स हैं कंप्लीटली डिस्कस्ड हैं। सो स्टार्ट नोटिंग एंड लिसन केयरफुली। बायोलॉजी में टर्म्स काफ़ी इंपॉर्टेंट होते हैं। अनटिल एंड अनलेस यू रिमेंबर द लैंग्वेज ऑफ मेडिकल इट विल बी टफ टू क्लियर इट। लाइक ग्लाइकोकैलिक्स, ग्लाइको का मतलब होता है स्वीट, शुगर और ग्लूकोज, कैलिक्स यानी कोट, यानी स्वीट शुगरी कोट. So very clear composition of glycocalyx is glycolipid or glycoproteins, and the lipid part makes it tough or protective, and the protein part helps it in reception or it helps in interaction. So we can understand here that the glycocalyx layer also enables the envelope to retain water. So it is slimy layer or can be also capsulated. Hence the function here written is protective, adhesive and reception. चलो भाई, पिछले वीडियो के एंड में मैंने एक सवाल पूछा था, व्हाट डिटरमाइंस द शेप ऑफ द सेल एंड प्रोवाइड्स अ स्ट्रॉन्ग स्ट्रक्चरल सपोर्ट टू प्रिवेंट द बैक्टीरियम फ्रॉम बर्स्टिंग और कोलैप्सिंग. सो कैन इट बी ग्लाइकोकैलिक्स? ऑब्वियसली नॉट. So it will be cell wall. So in NCERT, next to glycocalyx, that is the middle layer of the cell envelope, is the cell wall. A cell wall which is a semi-rigid casing that provides structural support and shape for the cell. So the next part of the cell envelope, the innermost part which touches the cytoplasmic area of the prokaryotic cell, is the plasma membrane, which is very similar, यानी वो बहुत मिलता जुलता है structurally with the eukaryotic model of cell membrane. That is, it is semi-permeable in nature. But whereas to remind you that eukaryotic model of cell membrane, or in the fluid mosaic model which we will be studying later on, is selectively permeable membrane. Now you can see the outer structure of the bacteria contains hair-like appendages which are called as fimbriae. This fimbriae helps in attachment to any surface for the bacteria, and if it is not associated with the movement or locomotion of the bacteria. Now if you'll see at the pilus, both the structures are almost similar. It is a modified fimbriae. This is hollow and elongated appendage which is very similar to the fimbriae, and it helps in transfer of the DNA to other cells, that is reproduction. Next important structure is the bacterial chromosome or nucleoid. इट इज़ हाईली कंडेंस्ड. सबसे पहली बात होती है, ये लॉन्ग होता है, सिंगल मॉलिक्यूल का बना होता है, लेकिन नो डाउट डबल स्ट्रैंडेड होते हैं और हेलिकल, बट दे आर सुपर कॉइल्ड इन नेचर. दे डू रिक्वायर्ड प्रोटीन्स बट नॉट हिस्टोन प्रोटीन्स टू गेट कंबाइंड टुगेदर इन अ कंडेंस्ड मैनर. अगर आमतौर पे एक बैक्टीरिया की साइज़ देखा जाए तो इट इज़ 2 to 3 माइक्रोमीटर्स इन लेंथ, बट वेरेज अ क्रोमोजोम इज़ नियर अबाउट 1400 यानी 1400 माइक्रोमीटर्स ऑफ लेंथ. जैसे आमतौर पे न्यूक्लियस के जो डीएनए होते हैं यानी जीनोमिक डीएनए होते हैं उनमें जीन्स प्रेजेंट होते हैं, तो बैक्टीरियल डीएनए के अंदर भी नियर अबाउट 3000 to 3500 जीन्स आर प्रेजेंट. If you will ask that what is the basic difference between a plasmid and a chromosomal DNA of a bacteria, then as I have discussed earlier that a chromosomal DNA requires a structural protein to bind together, but whereas a plasmid do not. Rest mesosome, flagellum and inclusion granules are discussed ahead. Let's move to the mesosomal part. ये एक important question है नीट के पॉइंट ऑफ व्यू से. outermost structure of the bacteria, that is cell wall, next to it is plasma membrane, and the infoldings of the plasma membrane, or we can say the extension of the plasma membrane, is the mesosome. It is formed in the three structures, that is vesicles, tubules and lamella. So the function of the mesosomes are described here. It helps in the formation of cell wall as it is close to it. It helps in DNA replication, distribution to daughter cells, respiration, secretion process to increase the surface area of the plasma membrane because it is a infolding, so it increases the surface area due to the infolding nature. And then it has enzymatic content which also help in respiration as well as content of enzyme. So this is noted from the NCERT. Next is chromatophore. Here you can see I have written it as exception. That means all the membranous extensions cannot only be called as mesosome, but whereas in case of cyanobacteria, these membranous extension are called as chromatophores. As the name suggests, chrome yani pigments, colored pigments. So it contains the colored pigments. This is what the structure is. Moving next, bacterial component. It is just a schematic diagram. I just want to notify the flagella present here. It has three basic parts, that is the basal body, hook and the filament, which helps in the locomotion. I have a question for you. Can you term that which type of bacteria is this? Particularly this part of the bacterial component? Yes, this is gram-negative bacteria because it contains two membrane, that is outer and inner membrane, which we discussed in the previous video. So moving next, we have the ribosome and the inclusion bodies. डिस्कस करने से पहले क्या आपको पता है कि राइबोसोम्स यूकेरियोटिक सेल में कहाँ प्रेजेंट होते हैं? ऑब्वियसली एंडोप्लाज्मिक रेटिकुलम, इसीलिए तो हम उनको रफ एंडोप्लाज्मिक रेटिकुलम भी कहते हैं. बल्कि राइबोसोम्स आर आल्सो एसोसिएटेड विद द वॉल ऑफ द न्यूक्लियर मेम्ब्रेन. Now, here in prokaryotes, that's a very minor question, that ribosomes are associated with the plasma membrane of the cell. Right, it was very interesting for me to find it out. Now, coming to the next point, that the prokaryotic ribosome, what will be the size of it? That is 15 nanometer by 20 nanometer. I just want to note you here that the sizes are expressed in nanometers. Yani we have moved down from micron size to nano size because we are discussing the organelles. Yes, obviously, if you're talking about the organism, it comes under micrometers, and if you're talking about the organelles, it comes under nanometers. The size gets on reducing. Whereas, just to remind you that the chromosomes length are more or larger than the organism size. It is obviously in same in case of humans as well. Right. So coming to the point that the ribosomes are of two subunits. One is 50s and is 30s. Do not get confused about it that 50 plus 30 is equal to 80s. Actually ऐसा होता नहीं है क्योंकि S Sydberg's coefficient होता है. जैसे कि मैंने आपको इसके बारे में पहले बताया कि सेटलमेंट कितना फास्टर एक मॉलिक्यूल सेटल होता है इन द सेंट्रीफ्यूज, सो दैट इज बीइंग रिप्रेजेंटेड बाय द S. So it is 50 and 30. So coming to the next point, that is polyribosomes. Here the image simply predicts that I think there are multiple ribosomes associated with an mRNA. That means the mRNA has brought some codes from the DNA or the genomic part, and the ribosome are doing their business. That is, वो लोग इसके कोड को ट्रांसलेट करने में लगे हुए हैं. यानी जो कोड्स हमारे हेरिडिटरी यूनिट जीनोमिक डीएनए के पास से आता है, वो मैसेंजर एमआरएनए उसको कैरी करके लेके आता है. साइमल्टेनियसली यहाँ पे राइबोसोम्स उन कोड्स को रीड करके ट्रांसलेट कर रहे हैं इनटू polypeptide proteins. So those are polypeptide proteins. Altogether, I want to say you that polyribosomes or polysomes, the ribosomes of a polysome translate the mRNA into proteins. And what is polysome? Group of ribosomes attached with the mRNA. So moving next to the inclusion bodies. So those are inclusion bodies. The images are a little bit tough looking, but don't worry. Let me explain it. Those are very simple. देखो, ये जो इंक्लूजन बॉडी है, द नेम इटसेल्फ इज कॉल्ड एज इंक्लूसिव. इंक्लूसिव का मतलब होता है विच इंक्लूड्स, और यानी ये जो इंक्लूजन बॉडीज़ होते हैं ये अपने में कुछ इंक्लूड करके रखते हैं. पहले मैं क्वेश्चन पार्ट को डिस्कस करना चाहता हूँ. सो यहाँ पे जो क्वेश्चन पूछा जाता है, दैट माइट बी दैट द इंक्लूजन बॉडीज आर मेम्ब्रेन और मेम्ब्रेनलेस. So the very clearly it is being noted that the inclusion bodies are not bounded by any membrane system and lie freely in the cytoplasm. So the example, what does it includes? It includes some reserve stored food materials, that is phosphate granules, cyanophycine granules and glycogen granules. Some other examples like the right side, that is I am showing a photosynthetic bacteria, that is cyanobacteria. It has some gas vacuoles, sorry. So these gas vacuoles are also can also be called as inclusion bodies present in the cyanobacteria. Those are forms of storage, but those both the organelles are the same. So here we can understand that inclusion bodies are nothing but reserve material in prokaryotic cells which store some cytoplasmic granules. Now in the left image you can see there are some inclusion bodies which are purple in color, and in the right image you can see there are some inclusion bodies in the green which is the cyanobacteria.
Thank you so much. Hope you find it useful. For any doubts and query, you can ask me in the comment section. If you like it, please share it. And the next video is very important. It's about eukaryotic cell organelles. Watch it.